Hello and welcome. Today, we are diving into one of the most incredible journeys our body takes on every single day, digestion. It's the amazing process that turns the food you eat into the fuel you need to live, and we're going to break down exactly how it all works.
I mean, have you ever really thought about that? How does a slice of pizza or sandwich or even an apple actually become the energy that lets you run or the building blocks that help you grow? Well, the answer is digestion, and that's precisely what we're here to figure out.
Okay, so here it is, the official scientific definition. The key words you've got to remember are large and insoluble, turning into small and soluble. Think about it like this. Your body can't absorb a whole brick, right? But it can definitely use the tiny grains of sand that make up the brick. Digestion is basically the process of smashing that brick into sand. So, here's our plan for the day. We'll kick things off with a big picture. Then, we'll take a grand tour through the digestive system. After that, we'll meet the microscopic powerhouses called enzymes, see how they work, and then cover absorption and the key lab practicals you need to know. And to finish, we'll wrap up with some top-notch exam takeaways. Right, let's start the journey. We're going to follow a piece of food from the very first bite all the way to the end of its trip, checking out each major stop along the way.
This is the main pathway, often called the alimentary canal. You can see it's a pretty long and winding road with several key organs that each play a really vital role. So let's start our tour right at the beginning.
First stop, the mouth. The journey kicks off here. Chewing is what we call mechanical digestion. You're physically breaking food into smaller pieces. But at the exact same time, chemical digestion starts. There's an enzyme in your saliva called amylase that immediately gets to work on any starches. Then, the food is pushed down the esophagus by these amazing muscular waves called peristalsis. Next, we arrive at the stomach. It's a pretty harsh environment in there, full of hydrochloric acid that kills off any nasty bacteria. And it's here that protease enzymes start breaking down proteins. Now, you'll also see these accessory organs, the liver, gallbladder, and pancreas. They aren't part of the main tube, but they produce absolutely essential chemicals for digestion. Their role is going to become super clear in just a minute. We've mentioned that word enzyme a few times now, but what actually are they? It's time to zoom right into the molecular level and meet the real heroes of digestion. Honestly, these are your body's secret weapons. Okay, here's the deal. An enzyme is a biological catalyst. Catalyst just means it speeds up a chemical reaction, and biological means it's made by a living thing. Specifically, it's made of protein. And the single most important thing about an enzyme is its unique 3D shape. That shape is everything. So how do they work? Well, scientists have a couple of models to explain it. The classic one is the lock and key model. It suggests the substrate, that's the molecule being broken down, fits perfectly into the enzyme's active site, just like a key fits into a lock. A more modern idea is the induced fit model, which suggests the active site is a bit more flexible and actually changes shape slightly to get a better grip. But in both models, the main idea is that the shape is incredibly specific. And this chart just shows how specific their needs really are. Enzymes are fussy, they only work well in their favorite conditions. You can see here that the protease in your stomach loves a superacidic pH of 2, but the enzymes down in your small intestine, they need a much more alkaline environment around pH 8. If the pH is wrong, they just stop working. Temperature is just as important. For us humans, the perfect or optimum temperature is our body temperature, 37 degrees Celsius. But if it gets too hot, the enzyme denatures. Now, this isn't just them taking a break, it's a permanent, irreversible change in the shape of that all-important active site. The enzyme is basically broken for good. It's like trying to unscramble an egg. Okay, now that we know what enzymes are and what conditions they need, let's meet the three main families of digestive enzymes that break down all our food. This table lays it out perfectly for us. There are three main types. First up is amylase, which tackles carbohydrates like starch and breaks them into simple sugars. Then you have protease, which works on proteins, turning them into their building blocks, amino acids. And finally, lipase breaks down lipids, or fats, into fatty acids and glycerol. See how the products are always those small soluble molecules our body can actually absorb? Now let's talk about bile. It's not an enzyme, but trust me, it's absolutely essential. First, it emulsifies fats. That's a fancy way of saying it breaks up large blobs of fat into tiny droplets. This massively increases the surface area for the lipase enzyme to attack. And second, it neutralizes all that acid from the stomach, creating the perfect alkaline pH for the enzymes in the small intestine. It's a true digestive hero. So, the food has been broken down into its tiny, soluble building blocks. Brilliant. But how does the body actually get them? Well, that brings us to the crucial step of absorption. And after that, we'll see how you can actually prove these nutrients are there when you're in the lab. The small intestine is an absorption superstar, and it is perfectly designed for the job. To start with, it has a massive surface area, thanks to millions of tiny finger-like folds called villi.
Its walls are also incredibly thin, just one cell thick, which makes for a very short diffusion path. And it has a fantastic blood supply to quickly whisk all the absorbed nutrients away, keeping the concentration gradient nice and steep.
Okay, time for some key lab work. To prove these nutrients are actually in our food, we use 4 simple tests. First, for starch, you add iodine solution. If it's there, it'll turn blue-black. For sugars, you add Benedict's solution and give it a bit of heat. It'll go from blue to a brick red color. For protein, adding beer red solution will turn it purple. And finally, for lipids, the emulsion test with ethanol and water will create a cloudy white layer.
Now, when you're doing a practical to investigate enzymes, you'll usually be testing one of 2 key factors, either temperature or pH. And the core principle of any fair test is to change only one thing at a time, your independent variable, while you keep everything else like concentrations and volumes exactly the same.
So the classic experiment uses the enzyme amylase to digest starch, and your goal is to time how long this takes. You'd set up your reaction and then take little samples at regular intervals, say every 30 seconds. You pop each sample into a drop of iodine on a spotting tile. At the start, it will turn blue-black. The moment the iodine no longer changes color, you know all the starch is gone and you can stop the timer.
All right, let's pull all of this together and really focus on the key questions that are going to help you absolutely nail your exams.
You will very likely be asked these 3 questions. First, why are enzymes specific? The answer is all about that unique active site shape which is complementary to only one substrate. Second, what is denaturation? That's the permanent change in the active site shape because of heat or pH. And finally, how does bile help? It emulsifies fat, creating a bigger surface area for lipase. Master these and you're golden.
If you remember just one thing about how enzymes work, please make it this. For an enzyme to do its job properly, it needs its optimum conditions. For your body, that means 37 degrees Celsius and the specific pH of whatever organ it's in. This is the golden rule of enzyme activity.
And we'll finish up with a little something to think about. The digestive system is a perfectly coordinated team. So what do you think would happen if just one of those players, one type of enzyme, like lipase or protease, were to denature and stop working? What would the knock-on effects be for the whole system and for your overall health? It's a great question to ponder.